Okay, so we're gonna start now. Thank you guys so much for coming to our class. Okay, so for this week, we're going to do Introduction to Macromolecules. So carbon is an atom that makes up and is found in organic molecules, which are the molecules that are found in living things. So carbon is extremely versatile because it has 4 valence electrons, which means that it has 4 electrons in its outermost shell. And this allows for carbon to form 4 bonds with a variety of atoms, including itself. And this makes it extremely versatile because it allows for carbon to form many different structures and compounds, including really long chains. And it can also form different things like rings and really short compounds and with singular atoms. So the first process that we're going to talk about is dehydration synthesis, which is when monomers are joined together, and this happens through the removal of a water, and it causes the formation of a bond between the 2 monomers. And on the other hand, hydrolysis is a process that breaks apart compounds into separate monomers, and this happens through the addition of a water. So our first macromolecule is carbohydrates. So its monomers are called monosaccharides and the polymers are called polysaccharides. And monomers make up polymers. So basically you can think of monomers as a puzzle piece and polymers as the actual puzzle. And carbohydrates are made out of the elements carbon, hydrogen, and oxygen. So you can remember CHO. And its function is it's the main source of energy. So you get it from like food such as bread, rice.
All right, and our second macromolecule is lipids. Now, lipids do many things in cells. They're used to store energy for long term, which is usually in the form of fat. They provide insulation for animals, and an example is that lipids provide a layer over fur or feathers for mammals and birds, making the water repellent. Now, lipids include fats, oils, wax, phosphides, and steroids. So fats and oils, a fat molecule is mainly made out of fatty acids and glycerol. Glycerol is an organic compound consisting of 3 carbon, 3 hydroxyl, and 5 hydrogens. And these fatty acids are usually made from a number of carbons ranging 4 to 36, as you can see on the slide. Fatty acids are called unsaturated when the hydrocarbon chain contains a double bond, and these unsaturated fats are usually liquid at room temperature and are called oils. So that's fun. When there are no double bonds, fatty acids are named saturated fatty acid, and this is because since no more hydrogen can be added to the carbon atoms of it. And trans fat is when hydrogen is present on different planes, and trans indicates the configuration of the molecule around the double bond. Now, phospholipids, they're composed of fatty acids attached, and there are 2 fatty acids forming diglycerol, and the 3rd carbon of the backbone is from a modified phosphate group. And then it is amphipathic, where the fatty acid chains cannot interact with water, while the phosphate-containing group can interact with water. The importance of these is that they are the outermost layer of animal cells and the plasma membrane. And now the 3rd, steroids. So unlike phosphates, fats, and oils, steroids have a fused ring structure. They are only grouped with them because they are hydrophilic and insoluble in water. Hydrophobic is when it is repelled by water and does not like it. Many steroids have OH functional groups, and cholesterol is the most common steroid and is mostly synthesized in the liver. Cholesterol is associated negatively, but it is essential to the function of the body.
The next macromolecule is proteins. So the monomer of proteins are amino acids. An amino acid consists of an amino group and a carboxyl group. And there are 20 different amino acids that you can use to make a protein. So this diagram on the right here shows the different amino acids. There are nonpolar amino
acids, polar amino acids, and there are basically different groups of amino acids that cause the structure of the protein to change based on their properties. When you join two amino acids using dehydration synthesis, you form polypeptides. And the bond that is formed between two amino acids are called peptide bonds. And there are 4 different levels of structure in proteins. The first level is the primary level, and this level is basically its primary structure where all of the amino acids are linked together by different bonds, and basically the sequence for each protein is determined by the gene encoding to the protein, and a change in a nucleotide sequence of the gene's coding protein may lead to a different amino acid being added to the growing polypeptide chain. The second structure is secondary structure, and this is where local folding of the polypeptide occurs in some regions, and the most common are alpha helix and beta pleated sheet structures. Both structures are like the alpha helix structure, the helix held in shape by hydrogen bonds, and they basically are held together by hydrogen bonds, which is important for a secondary structure. Tertiary structure is where chemical interactions using the R groups create a complex 3-dimensional structure of the protein. So because the nature of the R groups can be either polar, nonpolar, or hydrophilic, or charged, the protein will fold differently. So for example, sides that are positively charged and negatively charged will attract to each other. And or on the other hand, when two side groups both dislike water, so they're hydrophobic, they will stay in the core of the protein, and it will form this sort of like core in the middle because they don't like water, and that's really important for protein structure as well. Finally, quaternary structure is where weak interactions between the subunits help to stabilize the overall structure. For example, insulin has a combination of hydrogen bonds and disulfide bonds that cause it to be mostly clumped into a ball shape. And basically, it's just different like polypeptides that fold together into a big protein. And there are also different ways that proteins can be denatured or like broken down. So for like when you put a protein into high temperatures, a protein will be denatured. And if a protein is put into a pH that they are not like in nature, like the pH that they're not used to, they will also denature. And okay, the final macromolecule is nucleic acids. The monomer is nucleotides, and from the diagram you can see that a nucleotide is made up of a phosphate group, a pentose sugar, and a nitrogenous base. And the nucleic acids contain the elements carbon, hydrogen, oxygen, nitrogen, and phosphorus. So it would be CHON. The nucleotide, it's important because the phosphate group is negatively charged, and the phosphate group and the sugar form the phosphate sugar backbone of the nucleic acid. So it would be this like strand here and this part. And the nitrogenous base is the middle part, and it forms the ladders of the nucleic acid. So as you might know, nucleic acids are DNA and RNA, and both of these store genetic information and they encode information in general. But DNA is found in eukaryotic cells, and DNA is found in mostly like the actual gene, while RNA is used to code for proteins. So it's DNA to RNA to protein. And RNA is single-stranded, while DNA is double-stranded. And they also both have various other specialized functions, such as catalyzing reactions. Okay, so for the activity, we're going to do these two labs. Okay. So for detecting the type of macromolecule in certain samples, you can do different tests such as the iodine test, burette test, vinegar test, and litmus test. So our first sample A would be baking powder, and let's test this. All right, so as you can see, the baking powder is white. For litmus tests, it turned red, and then it also turned blue. For vinegar test, there were bubbles, and for burette test, it's blue, and for the iodine test, it was purple. So just kind of keep these things in mind, and then we'll let you guys guess what each test does to the sample. And let's do cornstarch. So cornstarch contains starch or carbohydrates. So for cornstarch, it looked the same as baking powder, except the vinegar did not bubble. Okay. I'm not sure why it's not working. So gelatin contains protein. wow, okay, so for gelatin, the blue litmus paper turned red, and the Bure test turned purple instead of blue, and the iodine test turned orange. Uh-oh. Okay. Let's see if we can refresh. Okay, I'm gonna stop share for a second so I can switch to my school account. All right, I think I forgot baking soda earlier. Let me do this. Is Okay, so can anyone guess what the Bure test does? Like, you can put it in the chat. Okay, I guess no one knows. So the Bure test turns purple in the presence of a protein. So if you see gelatin contains protein, so when you test it, it will turn purple. While all of the other samples do not contain protein. Can anyone guess what macromolecule the iodine test tests for? Yeah, okay, this is not working. Okay. The iodine test tests for starch. So if we test cornstarch again, it will turn purple when starch is present, and if starch is not present, it will stay orange. And well, the vinegar test tests for acids, and actually it tests for bases because vinegar is a weak acid. And the litmus test is used for testing both acids and bases. So if for red paper, it turns blue when the solution is basic, and the blue paper turns red when the solution is acidic. So if we were to test baking soda again, baking powder. Yeah, so as you can see, the red paper turned blue, so baking soda is basic. And for baking powder, since both stayed the same color, that means they are neutral, so like a pH of around 7. Alright, that's it for the activity. For homework, I will be posting a Quizlet where you can review the properties of each of the macromolecules. Thank you guys so much for coming. Do you have any questions? If not, you are free to leave. Thank you for coming. Bye.